to see.
Yep, and you can start now.
Okay, thank you. Thank you, everyone. And I am Filippo Nicolini again. I am from Italy, from the University of Bologna, and I am a PhD student who is now approaching actually the end of his PhD course. And today I'm very glad to be here to present my work, my main PhD work, which is about viral sex determination, of course, from a comparative genomic point of view. So first of all, let me just make some point clear about viral sex determination, because it's a very big topic, we can say. And basically, we have, during the last decades, we have started knowing something about viral sex determination, mainly regarding the fact that a mixture of environmental and genetic cues are involved in determining sex in vivals, and also that, for example, a polygenic system exists to determine maleness or femaleness. But basically, unfortunately, that's also that's everything that we know. So basically, I should have said what we don't know about viral sex determination. And just a moment, my slides are not working anymore. Okay, there we go. And that's why this little scallop is a little bit annoyed, just because everything that we know is really foggy and has a very patchy amount of knowledge. And also, I must say that overall, in the last years, thanks to the omics disciplines, we have fueled up the field a lot, mainly thanks to the availability of new sequencing data, both genomic and transcriptomics, but also proteomics. But again, we have a big but that is about the fact that everything, again, comes from very few species of socioeconomic importance. And everything, again, is really, we can say, yes, focused on a certain amount of steps in development or in viral anatomy, for example. So we are basically lacking the big picture in which to draw every study, in which to understand how genes have evolved and how the sex determination is changing through time and species. So in my PhD, just to make things a little bit easier, I just focused on the genetic part of sex determination. So I got rid of everything else regarding environment or sex chromosomes. And just because otherwise it would have been impossible, it would have taken me like 3 PhD or whatever. So I just focused again on the genetic part. And with this regard, what we know is that the genes that are likely involved in viral sex determination mainly belong to 3 big gene families, which are the DMRT, SOX, and FOX gene families. And again, that's all. I mean, we just know this about these genes because we are, again, lacking the evolutionary history, for example, of this gene family or their molecular evolution characteristics. But why do we have to know like the molecular evolution of phylogenetic history while studying sex determining genes? Well, because if we go back to the sex determining genes, to what we know about sex determining genes in animals, we can see that basically sex determining genes share some peculiar characteristics that are common through many different clades of animals. For example, regarding molecular evolution, again, sex determining genes tend to evolve faster, a bit more faster than other genes not involved in sex determination, or that, for example, they tend to evolve by duplication, by rapid duplication, also in very closely related species. So what I asked myself during my first months of the PhD was that if we know these features about sex determining genes in, for example, mammals or flies or nematodes, why shouldn't we use these characteristics also to understand which genes may be involved in sex determination in vivals? And that's basically what I did. So I wanted I started to retrieve, to build a comprehensive data set of the MRT, SOX, and FOX genes in bivalves, trying to cover as much as the taxonomic diversity as possible. So I employed both genomic data and transcriptomic data, and I built a pipeline to retrieve these genes in a reliable way, which means that the final matrix should have comprised only genes which was phylogenetic history and orthology relationship were certain and clear-cut. Otherwise, the genes would have just been discarded. Of course, this is very conservative, I understand, but what I wanted to have was just a set of true genes, just because, as I'm showing you in a while, the process of genome annotation and assembly in bivalves and in mollusks in general is very, very difficult, just because of the biology of the genomes themselves. So here you can see, I hope the bar on the top doesn't bother you. I don't know if you can see it or not, but anyway. Here you can see the matrix of the occurrence matrix of these genes, of the MRT, SOX, and FOX genes in bivalves. And basically, as I told you, there are many missing spots, and some genes are present in multiple copies, but this is just a reflection of how genome assemblies and annotations in bivalves are difficult to obtain. But overall, I'm not going to focus in much detail into this image, just because it would have taken just another like 12 minutes presentation or something like that. So here, what I just want to say is that now that I had all these genes, how could I understand if some of them were more likely involved in sex determination rather than others? So if we go back again to what I said before, if certain genes are involved in sex determinations, they should have a higher amino acid, a higher sequence divergence. So what I did is that I built the, here you can see on the top right, the distribution of amino acid divergence over across all the genes that I could retrieve as orthologous in bivalve genome and transcriptomes, which are more than 11,000, I guess. And just because I cannot see the number again for the Zoom bar, but whatever. And so now that I have built this distribution, I asked the dataset where my genes, the MRT, SOX, and FOX genes, fell in this distribution. What I obtained is that, of course, as you can see, the majority of the genes, the pink one of the MRT, SOX, and FOX gene families, fall in the range of the majority of genes from bivalve genomes, but some of them, which are the fuchsia one, show a higher amino acid divergence with respect to the others. And does these genes make any sense, like these highly diverse genes? Well, incredibly, yes, because, for example, the MRT1L and SOX-H have been already shown to be male bias when looking at their expression in gonads, for example, of many species, but also they have been included in the working model of sex determination for the Pacific oyster. Here you can see them. And in particular, they have been included in the male sex determining cascade. I want you also to focus on these genes, which is FOXL2, which is present, of course, in my analysis. And in this case, it doesn't show a higher amino acid divergence, but this just can be a reflection of the fact that maybe FOXL2 is not present at the same level of the sex determining gene regulatory network. So maybe it's just a basal effector. Or maybe there is something else going on with these genes, which is maybe, for example, that this genes is involved in sex determination just in certain species and not widely across bivalves. But at this point, again, I was like, yes, the signal is here, but I'm not very convinced about it because this signal can just derive from something else about these genes or something that is very some other biological phenomenon. So what I did was to rerun the pipeline again but in mammals, in which basically the genome annotation and the functional characterization of gene is much more solid than the one in bivalves. And here, as you can see again, this is the matrix of genes, which is far more complete, just because, as I told you, the genomic resources for mammals are much more reliable and much more curated up to now. And again, I draw the distribution, the amino acid distribution that you can see on the left of the amino acid divergence of genes of orthologous genes in mammals. And then I looked at the genes which from the DMRT, SOX, and FOX gene families, in which part of this distribution they fell. Again, the majority of them just fall in the range of the majority of genes from mammal genomes, but one of them, which is SRY, stood up from the other. And what is SRY? It is the male sex determinant in mouse. So basically here we can see again the same pattern that we saw in bivalves. So apparently, if a gene maybe is the male sex determinant or likely at the very top of the sex determining cascade, it can show a very higher amino acid divergence with respect to the other. Again, FOXL2 is present also in the mammal sex determining cascade, but again here is just it just fall in the range of the median range over the genes. Now I didn't highlight it. Oh, here it is. So again, this show how maybe also the position on the cascade is important to determine the sequence evolution and the molecular evolution of these genes. So to wrap up everything, in the last slide, in the last 10 minutes, 12 minutes, I don't know, I hope that I convince you that drawing the evolutionary framework and the comparative framework for studying sex determination in a certain group of animals is very important to understand how the genes basically evolves, in particular for those clades of animals or of organisms in general, of which we don't know much and we are still learning a lot. And of course, this doesn't this is not true just for sex determination, but for any basically biological process. And also for what concerns biosex determination in particular, the MRT1L and SOX-H, again, have been shown also by molecular evolution analysis that they are very good candidates to focus on and to just I don't have the word to study in more detail, also from a functional point of view. Also has shown by the pipeline that has been run on mammals. So for today, that's all. I think that everything was more or less clear. I hope that you enjoyed my presentation, and you're very welcome to get in touch with me. Just visit my website if you want to, I don't know, write an email or whatever, or just post a question here. And I may happen to be looking for a postdoc. So if anyone knows about anyone who is hiring or who has some open position, I would be happy to know about that. So thank you again to the organizer for letting me present to this very, very nice Congress. Thank you again.
All right, so we're going to have you stop sharing your screen, and Joanna, you can start sharing yours, but we've still got a minute in case anyone has a quick question for Filippo. All right, with no one asking a question, I am going to ask you, do you have a reason why you suspect that there are so many more duplications within that set of bivalve genes that you showed us versus the mammals?
Oh, yes. Probably the fact is that those duplications were actually in a very interesting part of the bivalve tree of life, which is the muscle, like the Mediterranean muscle, the California muscle, and all that part, which actually have a highly dynamic genomes, in the sense that even within species, you can see very different gene copy number variation. So probably it's something related to that. the fact that just
All right, I'm going to thank you for that, but cut you off here because it's time for Joanna to start.
Sure.
And Joanna, you can start whenever you're ready.
All right, so thank you. I'm going to be talking about the genetic code and how it evolved and how we know. So this is the genetic code. You've probably all seen it before. How it evolved is a big mystery. It is obviously very complicated. We presume it did not evolve all at once. It must, like all complicated things, have evolved in stages. So what are those stages? 20 amino acids is quite a lot. Probably the genetic code started out with fewer amino acids and then more amino acids got added to it. So sort of a key part of the stages is what order were the amino acids added to it. So there's, in the field, the current state of the art is to take 40 lines of evidence, each of them pretty bad, and to look for statistical consensus that the 40 are all pointing in the same degree, kind of average them, and then say that's the order of recruitment. And those, to give you an example of sort of how some of them are pretty bad, one of the, quite a few of them have to do with various estimates of whether an amino acid could be present abiotically, other than, just from chemical processes. There are some problems with that. One problem is that one of the sources of evidence is something called the Urey-Miller experiment, where they kind of tried to make amino acids in the test tube. They did not add sulfur. Methionine and cysteine have sulfur in them. You cannot make sulfur if you do, there is conservation of matter here. And so, that leads automatically to methionine and cysteine being late, even though there might have been sulfur on an early Earth. It's also the case that the genetic code probably evolved in an organism that already had complex RNA metabolism. What was available abiotically in the primordial soup does not necessarily represent what early life had to work with at the time the genetic code was being constructed. So we wanted to find a more biological rather than chemical way of figuring out the order of recruitment. And to exploit that, we exploit the fact that not all genes go back to the primordial soup. In fact, most of them have been born since then. Genes are being born de novo from junk DNA or alternative reading frames all the time. So if we could identify the minority of genes that are truly, truly ancient, we expect those to have more of the really old amino acids and fewer of the later added amino acids. So essentially, we want to have trees like this, where, we can see that there's branches between bacteria and archaea, and they're nice long branches, and you can figure out that the last universal common ancestor was here, or in some cases that, we can go even further back, in the case of duplications, further in evolutionary history than the last universal common ancestor. And we don't do this with genes because a gene is like the car and it is made up of domains, which are like the wheels. And we care about the age of the wheel and what amino acids went into building it, not the age of the car. And different genes today have these different components that were added and subtracted and duplicated and so on during evolution. So we focus on the PFAM domains. PFAM's just the classification we get that from. So for people who are phylogeneticists, we make domain trees. Brief thing of our methods. We make domain trees, we reconcile them with what aren't very good species trees, but it still helps to reconcile. We do midpoint routing procedure where we ensure robustness to outliers. And then what we do is we look for things that are clearly horizontal gene transfer on the tips, and we prune them out. And after we've pruned them, we're then able to identify things that go back to Luca via this sort of long branch separation between archaea and bacteria and the coherence of that. So previous analysis of what was in Luca is this Weiss et al. Paper, which had whole genes. This is our work, which is for PFAMs. And I think the main point here is we have more stuff. You have more to work with. And then by looking at some of the case studies of what we include and what we don't include and what they did and what they didn't, we're pretty sure ours are also of higher quality as well as higher volume. We also do what are called clans of PFAMs, some very ancient things we know from their very similar structures and doing structural alignments that are, sorry, I have a dog wanting to join, that they are in fact related. So we have multiple PFAMs in the same. And so that leads to even more ancient stuff when we take it at the clan level, because that's taking advantage of annotation. That something genuinely is more ancient. And yeah. So looking at these clans that go back to Luca, what we find is there is a correlation between what people believed on the y-axis was the order of recruitment and what we see, which is how much of the amino acid is used in these Luca clans compared to other pretty old but not as old protein domains. There are exceptions, though. There are 3 outliers to this relationship that were not previously believed to be abiotically available. And they include the cysteine and methionine that have sulfur in them, which explains that. And histidine is interesting because if you look at the structure of it, it looks a lot like a purine. So it's not very abiotically available, but to an organism that did a lot of RNA-based life, it could well have been highly available. Histidine is actually really, really, really important. It is by far the best amino acid at binding transition metals that are key to catalysis. Cysteine is kind of the second best. So we have this story of really important metal binding functions in early life. Also, the genetic code itself, you know, core to these proteins called synthetases, which attach very specifically the amino acids onto the right tRNA. And they have a motif called high, which has 2 histidines in it. So it's pretty hard to imagine how you could get any kind of genetic code if you didn't have histidine. So this actually makes sense. The 3 exceptions that we found all kind of feel like exceptions that prove the rule. Another thing I just want to point out is that tryptophan was last, by a very, very large margin. It is very strongly depleted in things that are very ancient. And indeed, if we look at the synthetase that adds tryptophan, we find out it wasn't even in Luca. So this is showing it as an unrooted tree just to illustrate our rooting process, that our root ends up on this black branch here, which goes between, basically says, oh, okay, there was a tyrosine synthetase in Luca, and then the archaeal tyrosine synthetase duplicated and diverged into tryptophan synthetase, which was then transferred to bacteria and moved throughout that. This was not previously known because people had done whole gene alignments and we did a domain alignment. There is a second domain that is unique to tyrosine synthetase and is deleted in tryptophan synthetase. And if you align apples and oranges and you have a whole lot of things present in 2 sequences and missing in the other 2, there'll be an attraction between those 2 branches that doesn't represent the real evolutionary relationships. And another thing is, if you actually look at how tryptophan is synthesized, a key enzyme of that actually came not in archaea, but in bacteria. Overall, we get this picture of tryptophan being added really late as the 20th amino acid. So far, I've actually only told you about the clans that are on the right that sort of existed in 1 copy in Luca. Now I want to turn to the even more ancient ones that already existed in at least 2 copies in Luca. And for these, we actually see absolutely no relationship with the order of recruitment that was previously believed. Although we do see a relationship on the x-axis of what the enrichments we see in Luca and on the y-axis of these even older, what we're calling pre-Luca protein clans. There are some statistical differences, though. Those are that it is less depleted for tryptophan, less depleted for tyrosine, and is actually enriched rather than depleted for phenylalanine. So an interesting interpretation here is, at least in the laboratory, tryptophan, tyrosine, and methionine are the 3 amino acids that can provide antioxidant protection. Now, of course, there wasn't any oxygen on the early Earth to speak of, but there may have been a lot of sulfur. And reactive sulfur species are, in fact, oxidants. So you can think of these amino acids also as anti-sulfidants as much as antioxidants, oxidants being a modern thing. And so you could say, okay, maybe all three of these were important once. And then when the sulfur went away, we didn't need them as much. But when the oxygen came back, they got used a whole lot more. So we sort of but we still thought tryptophan is this big, expensive thing. It was really surprising that there was so much of it in these really ancient things. So we sort of said, okay, is it a bug? Let's look at which one has the most tryptophan. Let's just check that, this isn't nonsense. And it was a synthetase again. It was the synthetase domain that went into leucine, isoleucine, and valine, and it's sort of 4% tryptophan, which is really weird. So this obviously predates the modern genetic code by a lot, and yet it has not just a lot of tryptophan, but there are tryptophan sites in the active pocket, 100% conserved, that are actually, key to the shape of that pocket, which allows its discrimination of its core role of being able to tell the different amino acids apart. And hence, this is the core of the genetic code. So, the way I look at it is just because tryptophan was the last addition to our genetic code, that doesn't mean it wasn't around before that. I should note that in this leucine, isoleucine, valine, it's not just that you've got conserved tryptophan sites, you've also got conserved isoleucine, leucine, and valine sites, which go back prior to, this is the evolution of the protein domain that does the discrimination between isoleucine, leucine, and valine. And yet, conserved isoleucine, leucine, and valine already existed before that. So basically, what we're seeing is glimpses of information about prior to our genetic code. We're getting glimpses of information into an era in which there were competing genetic codes. So to sum up, if we compare the order of recruitment of the if we compare the use of amino acids in LUCA as proteins to ancient stuff that's not that ancient, we learn about the order of recruitment. We learn that there was probably a lot of sulfur around the time that this, our genetic code was getting going, that metal binding was super important, and that tryptophan was super late, actually, after the last universal common ancestor in Archaea. But if you go back to earlier things, more tryptophan, more tyrosine, more phenylalanine, that may reflect different usages in competing codes that came earlier, and also including a threat from reactive sulfur species. Okay, and thank you to the people. This is the most of this work is Sausan Webbies, who's in the wrong time zone to present. So I'm presenting her work. Andrew did some stuff too, as did these other collaborators. And thank you for your time.
All right, and we've got 2 minutes until the next person. So Joanna certainly has time to answer some questions. And that requires us to have questions. So could you just briefly tell us what some of these other lines of evidence were that you didn't get into as much earlier?
Which, the 40 criteria?
Yeah, like just general, what are they trying to look at?
Like, how tight is the codon-anticodon binding? How common is the amino acid in its use today? How big and complex is the thing? Not a one of the 40 is particularly persuasive. The evidence really came from some degree, but it was a pretty crazy paper, because they actually started with more than 40 and then looked for concordance and then threw out the metrics that didn't have enough concordance. So it was worse. It was actually pretty bad.
I want to follow up that question. I wonder, did you check and see if your new determination matches more or less with some of those different criteria? Because that might be cool to look at to see which of those are actually robust.
Yeah, I think good question. We've thought about doing this. I think we just haven't at this point.
All right, so I'm going to
Yeah.
cut you off there because it's just time for Jordan to start, but thank you, Joanna.
Okay, hi everyone. My name is Jordan Gossett. I'm a PhD candidate at the University of Hawaii at Manoa, working in Dr. Becky Chong's lab. And today I'm going to talk to you about some of my work uncovering cryptic diversity in cave-adapted plant hoppers. So my work is focused on Hawaii Island, which is also known as the Big Island of Hawaii. It's the youngest island in the archipelago. It's only estimated to be around 500,000 years old. And it's made up of 5 volcanoes, 3 of which are still active today: Hualalai, Mauna Loa, and Kilauea. And Kilauea and Mauna Loa experience pretty frequent volcanic eruptions. This is actually an image from the air of the most recent volcanic eruption from the Mauna Loa volcano in 2022. And these lava flows happen all the time. And when they cool, they form a really hard outer shell while the interior of that flow continues to flow out into the ocean. And they create these hollow cylindrical structures in the subterranean environment that we call lava tube caves. So these are 2 images of entrances into lava tube caves standing on the surface looking down. And you can imagine that as we have lava flows occurring on top of one another from different volcanoes throughout many years of the history of the island, we get tons of lava tube caves. And this image on the left of the island shows us within the more recent geologic past, some of the different lava flows that have occurred from different volcanoes. All of the different colors representing lava flows that have occurred on different years and timescales. And within each of these different colors, you can imagine we have sometimes within 50 to 100 different lava tube caves that are all sitting on top of one another, beside one another, and intersecting one another. So this is a really fragmented network in the subterranean system of the Big Island. And so just looking at those images, you might notice that the lava tube caves look kind of barren, but they're actually not. They harbor really diverse troglobiont or communities of cave-adapted organisms. And there are some examples shown here. These are all organisms whose surface ancestors at some point made their way into the caves, and that founding population continued to spread. And eventually we have these cave-adapted lineages popping up. And so you might be wondering what's sustaining these abundant communities underground, and that would be surface vegetation. So we get these barren lava fields after an eruption, and eventually ecological succession takes over, and we get trees and shrubs moving into the environment. And they project their roots from the surface into the caves below to obtain moisture. So this is an image of my advisor and myself within a lava tube cave, and you can see hanging down from the ceiling is a pretty extensive root curtain from a tree located on the surface. These roots into the cave habitat are the only source of primary nutrition. And so the important link between this source of primary nutrition and the rest of the cave-adapted community are cave-adapted plant hoppers. So these guys are super abundant in the caves. They're the only primary consumer in this system. So they feed directly from the sap of tree and shrub roots that hang from the ceiling. And they use nutritional endosymbionts to turn that nutrient poor sap into essential amino acids and vitamins and so forth. And then pretty much everything else, all of the other species living in the caves feed directly on plant hoppers as their main nutrient source. And so for that reason, cave-adapted plant hoppers are known as a keystone species or a keystone group in this ecosystem. So it's really important that we learn about their population distributions and their biodiversity. And we don't know a whole lot at this point. If you go into the caves, you'll see cave-adapted plant hoppers kind of everywhere hanging out on these roots. They all look very similar to this image that you see here. This is a nymph. But the main species that we find that's the most abundant is Olearis polyphemus. And there's been 2 studies done on polyphemus assessing their biodiversity underground from different angles. The first would be this study by Hawk and Howarth in 1993. So you can imagine in a cave adapted system where we have total darkness, plant hoppers fully cave adapted, they have lost their ability to see. They rely on acoustic vibrational mating signals to locate and identify mates. And so it's really important in this system. So what Hawk and Howarth did was they recorded acoustic mating calls from plant hoppers from these different cave sites that you can see on the left. And what they found was there was significant differences between the mating calls of plant hoppers collected from different caves. So there's really strong phenotypic variation. And what's more is when they went to mate these plant hoppers in the field and in a lab setting, they found really strict assortative mating. So plant hoppers from Mackenzie Park will not recognize or mate with plant hoppers from Pahoa Cave, for example. And that stands true kind of across their sampling sites. And then in 2013, Wessel and colleagues built upon this work with some genetic diversity work where they were assessing CO1 sequences between plant hoppers collected from these same cave sites. And what they found was there was pretty strong genetic diversity in the CO1 sequences. This combined with the phenotypic variation and the assortative mating patterns led them to hypothesize that Olearis polyphemus is likely a species complex rather than just a singular species of plant hopper underground. And so this is kind of the groundwork of what we know up until this point about cave adapted plant hoppers. So that's where my objectives come into play. This is a portion of my dissertation work where I was hoping to understand the Olearis polyphemus species complex on a broader island-wide scale. So those studies were done with limited sampling sites across the island. We now have access to quite a few more sampling sites through permitting, especially in that southern Mauna Loa region. And I also am using nuclear genomic data rather than amplicon sequencing to assess the genetic diversity. And then I also wanted to quantify gene flow between groups of plant hoppers to identify potential barriers to gene flow so that we can start to tease apart this species complex and understand where those physical boundaries between species might exist. And so to do that, I collected 320 cave-adapted and surface plant hoppers from the island of Hawaii. Of course, I've collected plenty of Olearis polyphemus, but then we also collected a few Olearis filicola samples, which you can see in the image on the bottom. Those Olearis filicola is a surface relative of polyphemus, and so we were using that more or less as an outgroup for this study. I did CO1 barcoding on all samples just to confirm species identity, and then I have an assembly of 13,000 SNPs for 100 of those 320 samples. And this image on the right shows you some of the lava flows from the different volcanoes that we sampled from. So first things first, we ran a maximum likelihood tree with the 13,000 SNPs, and I'll kind of walk through the different portions of the tree here. So this top outgroup was our Olearis filicola outgroup, and the rest of the tips on the tree are representing Olearis polyphemus samples from across the island, and that's the results that I'll focus on today. So I'll walk you through, just going to highlight some of these clades so you can see where they were sampled from across the island. First top group in yellow were collected from this northern QK5 flow in the Mauna Loa volcanic region. That flow is around 200 years old. We also have a flow within the Kilauea region, the QP4 flow, that's around 750 years old, represented in gray. We have these 2 clades that were collected from the QK3 flow in the southern Mauna Loa region. That's around 1,500 years old. And then a monophyletic clade collected from Hualalai flow that was around 3,000 years old. And so I think the big pattern that jumped out to me here was that we have from the middle northern portion of the island, all of those flows form more or less nice monophyletic clades, whereas those Mauna Loa plant hoppers collected from QK3 form the polyphyletic clade. And so this indicates there's some sort of genetic divergence going on with plant hoppers collected from this individual flow that we need to investigate further. And so that's what I did using admixture and a PCA analysis. And you can see the results of the admixture here. We've got Ks 3 through 5. And then on the bottom are the different chunks of individuals collected from each flow. And if we look just broadly at this, we see there's not a ton of admixture really at all. There's two main groups. There's either the blue group or the orange group, especially within QK3, and you're kind of one group or the other. And we see the same for some of the other flows. This is also represented in our PCA analysis, where QK3 represents two genetic clusters, one cluster kind of grouping more with those northern Mauna Loa Kilauea flows, and one cluster grouping more with our Hualalai individuals. And so here we see evidence of two distinct Mauna Loa lineages that potentially originated from other volcanic regions coexisting together. And I wanted to take a little bit deeper dive look into some of the individual caves from this QK3 flow. So I have 3 caves here: Cordwinder, Wilson, and Maelstrom. And these represent individuals that were collected from, we're talking the same root within a cave. And you can see here, individuals collected side by side are showing not a lot of gene flow with one another. There's still not a lot of admixture. There's still these distinct groups, even within individual cave systems. And so again, we're seeing those divergent lineages of the Olearis polyphemus complex coexisting with one another. And there's a lack of gene flow even on this really super fine scale in this system. And so I'm kind of in the midst of this right now, but overall, some conclusions are that we see that lack of admixture across the island within the Mauna Loa QK3 and individual caves. There's a potential for multiple lineages of the Olearis polyphemus species complex coexisting with that strong assortative mating. So we're seeing that now from a genetic standpoint rather than just the mating trials evidence. Although this is all great, but the species complex is far from being resolved. That's kind of what I'm working on at the moment, is teasing apart all of this genetic diversity and running different analyses to try to understand truly what's going on. And so my next steps to attempt to figure this out are running species delimitation models like BPP and some things in beasts to try to determine, what species boundaries are in terms of all of this data, phenotypic, behavioral, and genetic that we have accumulated. And then I'm also in the middle of running some introduction route simulations. And so we know that cave adapted plant hoppers are a result of an invasion from some surface ancestor into the caves that became cave adapted over time. And so what we want to figure out is how many surface invasions were there? Was there one surface invasion where plant hoppers have kind of, migrated and diverged underground, or are these divergent plant hoppers the result of several different surface invasions that are coming into contact with each other beneath the surface? So with that, I have my land acknowledgement, and I'd like to thank, of course, my lab and all of my funding sources, and take any questions if you have them.
And we certainly have time for a question or two. All right, so I'm actually going to ask a question if no one else is going to. So I was curious, when you showed your set of the individuals living in the cave, looked like everything was pretty much within arthropoda or something in there, but clearly these are relatively wide mouth caves that you and your advisor could get in. Do you not have any like bats or birds coming in to try and eat any of the insects?
We do. We see those kind of right at the entrance is where you'll see like some mammals and things kind of hanging out. But once you get into deep cave, like total darkness zones, the root curtains are so patchy, like you might be walking for miles before you actually see a root curtain, and then you might see a ton of roots and a ton of insects. So within the deep caves, it's super patchy and not a lot going on besides those arthropods.
That makes a lot of sense. Thank you. So Wei, do you want to now? Oh, I'm sorry, Jigar. Can we come back to you after Wei gives his talk, because we'll have some time left over before the session ends, if you're okay with that.
Yeah, that works for me.
All right, so you should be able to start whenever you want.
Okay, hello everyone. This is Wei Li. I'm a postdoc working with Gavin Sharrock and Stanford University. So Sharrock Lab has previously developed a molecular barcoding system for lineage tracing. And today I would like to talk about a synthetic biology tool that we put together recently, which we use the positioning barcode to trace the physical locations of those clonal arrays. Hopefully at the end of this talk, I would like to convince you it can be used to generates sequence verified and well-balanced libraries. I'm still trying to organize my windows on my side, so let me do it very quickly. All right. So here, like I think during the in-person session of the evolution meeting this year, there is going to be a symposium aims to discuss different methods to measure the fitness effect. So I think one classic way to measure the fitness effect is to use the polymorphism data that we collected from the natural environments by sequencing. And also in the past decade, I think technology advances also allow us to do this large-scale measurements of fitness effect in the lab. And this is related to what I'm talking about today. And here I just list a few of the well-known names of this approach, for example, the deep mutational scanning and massively parallel reporter assays. So for this whole method to work, I think it depends on initial construction of the variant libraries. And ideally, the relative frequency of each of the variants should be equal. So later, the selection can be applied, which means, for example, we put antibiotic drug treatment or temperature shift. And later, the frequency change of this variant can be used to calculate the fitness effect of each of the variants. And I think one crucial component of this whole approach is to generate the very initial variant library pools. And so far, we actually have a lot of methods to generate the input variance libraries, but I would say it's more or less like can introduce some sequence errors or unbalanced. And I think these issues are not trivial if we consider how the whole method is working. For example, if the one of the beneficial mutant, for example, the first one is systematically underrepresented in the initial pool. So there can be two issues, in my opinion. First one, it can, even though it's beneficial, it can go extinct because the initial frequency is so low. This can go extinct due to random genetic drift. And also there can be some sequencing cost considerations because it's so underrepresented. We need to generate a lot of more sequence rates in order to accurately estimate the relative frequency of this variant in the initial pool. And plus this, and there can be sequence errors during the library constructions, which introduce some unexpected variant in the pool, which can cause like another layer of complexity when we try to understand the distribution of variant frequencies. So I think this really motivates me to figure out an approach to do the sequence validated and also well-balanced variant libraries. So there are a couple methods available at this moment. For example, this dial-out PCR approach, which uses the pooled sequencing and also the target-specific PCR, which allows us to, for example, we can pick up the variant we wanted and pull them together. And another approach is this called micro-wheel-based multiplex sequencing, which in each of the wheels, we need to design the unique barcode to do the PCR, and later we do the pooled sequencing. But in my opinion, both of these methods are not, it's very difficult to scale up because as you can tell maybe already, a lot of like huge number of unique pair of primers need to be designed. And also there can be off-target effect. And also there are limitations from the reagent cost. So I think this also like motivates us to figure out approach by ourselves. And today I would like to talk about a method we propose here, which rely on this bacterial conjugation and recombination. And the pipeline of our method is the following. For example, like we can firstly generate any type of variant libraries. So the diagram here indicates the initial libraries was generated by this microarray synthesis. But later in my another cases, I will show you, I think it can be any type of variant libraries generated. We can onboard them into our system. So after we got these initial variants, we clone everything into our donor vector and also the donor cell, which is an engineered E. coli. And later we pick up random clones and put them onto a random array. So on each of this array, we have one, on each of the position, we have one unique variant, but we don't know the sequence information and we don't know where it is. But later we can mate the random array clones with the positioning barcode. And this positioning barcode plate is something we prepared before doing this mating. And on each of the position of these positioning barcode arrays, there is a positioning barcode. We know the sequence information and we know where it is on this array plate. So during this conjugation, what happens during on the molecular level is this unknown variant will be recombined with our positioning barcode. So they can physically associate with each other. So later we can do a pooled sequencing, no matter it's a short read sequencing or nanopool PacBio sequencing, we can use the known information of the positioning barcode to know the sequence identity and position of the variant on the plate. So there are a couple of features I really like about our method. Firstly, it's a simplicity in execution, because once we get this donor array, we just simply mix our plates together for the bacterial conjugation, and then later we do everything in a pool. We do pooled, for example, plasmid extraction and pooled sequencing. And secondly, I think it's pretty cheap. And in this method, we provide two modes of execution. One is what we call low throughput, because in some, for example, individual labs, if we are only interested to construct the variants on, for example, multiple 96-well plates, we can use the pin to do the manual bacterial mating and conjugation. If we want to scale up, we have, for example, automatic colony picker and some other robotic handling system to do the bacteria mating. Here's the cost estimates include both the labor cost and reagent cost, and they are one order of magnitude lower than the current methods. And lastly, I would like to say the turnaround time is pretty short, because the barcoding procedure really take over overnight. And by the end of the next day, for example, if we use nanopool, we can start to generate data by the second day, and we can do the data analysis. So in practice, we have followed this pipeline, and we sequence already more than 45,000 plasmids containing different variants already. And I think it works pretty well. And these are some of the three features I want to highlight here, but I think I haven't convinced you this method is accurate and efficient enough. So I think we definitely need to do that. So to demonstrate these capabilities, we do the following experiment. We use unknown variant DNA plate and mate them with 8 different kinds of positioning barcode. Because we mate them 8 times, which means we sequence them 8 times. That means we can cross-check the results to evaluate the accuracy. And the result shows here, by even one time of mating, one time of sequencing, we can get 100% accuracy. And because we mating 8 times, we can ask another question about the efficiency, such as by mating how many times we can recover 100% of the information from the unknown plate. And the result indicates even by one round of mating and sequencing, we can recover more than 98% of the unknown information from the unknown DNA variant plate. So after this kind of proof of principle and check this efficiency and accuracy, we have an interesting application to use our BPS system to sequence validate and rebalance those open reading frame libraries. And upstream to our BPS system, we rely on this people called inversion probe approach to capture the genomic capture the open reading frame from the genomic samples from environmental samples. And this approach was recently like adapted by Bijou Group from Rutgers. And I like to use this diagram a lot because I think it's straightforward. So this probe just serves as a nozzle to capture and specifically bind to the open reading frames that we are interested in and to clone them and capture them. And besides the high throughput nature of this approach, they call the nozzle approach. I think another highlight here is it can capture some unsynthesizable variants, which also extend our capability to test the fitness effect of these variants in the lab. But there, of course, are some disadvantages of this approach. For example, there can be off-target DNA captured in this pool, and there can be some sequence error because there are a lot of like amplification procedures, and there can be unbalanced abundance introduced. So after we like digest this approach, we think, oh, it's really can be a nice place, nice application for the BPS to perform. So we collaborate with Bijou's group, and the first 3 parts just their procedures, like they capture everything from the environmental samples from the genomic DNA. And later, what we can do to onboard everything, to onboard the off library into our system is pretty simple. We just order 1 pair of primers, do the amplification, to the digestion of the plasmid, and clone everything into our system. So after sequencing, here is our result. We notice like the 27% of those clones we pick up are some mixed clones. So these mixed clones can result from, for example, code transformation or some errors I made when I tried to pick up a single clone. And for most of them, they are pure clones. And during this, like 30%, around 30% of them are sequence perfect offs that we can use to construct our off variant libraries. And there are large portion of them are offs with errors. They are mostly like 1 to 2 nucleotide substitutions from the reference. And we have additional evidence to show that these errors are not introduced by our BPS, but from our previous upstream procedures, because there are a lot of cloning steps performed during the protocol. And of course, we can, even though the off-target DNA is a small portion, but we can accurately like to filter them out. So later, after we get these sequence perfect offs, we can just pick up those clones and rearrange them and pull them together. Here, I would like to show the uniformity of this rebalanced pool before and after the pooling. So the axis here just shows just list all of the offs we deal with here according to their abundance, rank from the most abundant one to the least abundant ones. And the y-axis just the frequency. As you can see here, the original pool before the passing, it really dominates by a few of the off version. But I think after the BPS, after sequence validation and after repooling, and the uniformity is substantially improved. And so ideally, like if it's a uniform, like there we expect to see a flat line, but I think this is like compared to the original distribution, it's significantly improved. To do the repooling, we try to pull the variants from either the agar or from liquid, and both of them works quite well. So to summarize everything, today I just want to show you this high throughput sequencing verification and rebalancing system. We call it bacterial positioning system, or BPS. It can be used to sequence verify thousands or tens of thousands plasmid clones contain different variants with a single mini prep and pooled sequencing. And we also think it can be widely compatible to various methods to generate sequence verified and balanced variant library pools. And lastly, I would like to point out this method so far is not perfect, because if, for example, in the situation, if the initial pool is very skewed, we need to like pick up a huge number of single clones in order to capture those rare variants. So I think that limits our capability to recover all the DNA variants in the pool. But I think that's a future direction we would like to work on. Lastly, I would like to thank my advisors, Sasha and Gavin, and also co-authors, collaborators, and funding source. And if you are interested, you are welcome to check our recent publication, and hopefully we can have some interesting application out of that. Thank you.
All right, so we are just at 4:30, but we only have 4 speakers in this session. So I'm perfectly happy to keep this open for longer if people have questions for any of the speakers. All right, you should be able to unmute now.
So I have first of all, great job with organizing this. I had a question to the previous speaker regarding.
She is not in the session any longer.
Oh, okay, sorry.
So that would make it
That's
harder to
okay.
ask her a question. You might be able to
But-
track her down later.
Was it for me or?
Oh, sir, you. I'm sorry.
Oh,
I
yeah,
don't know. I don't
yeah.
know.
My
Yeah,
mistake.
sorry, I just missed your name. So I had a question that you said that you use the sequencing, I think the next gen sequencing data for CO1 markers, and you did not use the amplicon sequence data.
Yeah, so we did DD-RAD library prep of SNPs throughout the genome for
Right.
next gen. Yeah. We have
Right.
CO1 as well. But I was just showing the SNP data.
Okay, so did you look at the potential contamination of nuclear mitochondrial sequences of COI? Like the portions of the mitochondrial sequence or the mitochondrial gene of CO1, which has integrated, because that is one of my like projects, and my model system is Daphne, and I was able to find nuclear mitochondrial sequence for every mitochondrial gene, and therefore I had to segregate those reads and separate and like remove them out bioinformatically because they could give you a sense of a false, like a false positive SNP.
Oh yeah, no, that definitely makes sense. As of now, no, we haven't checked for that.
Okay.
But have you given a talk yet? Or if you're giving one, I'd love to come check it out. And
I'm actually in Montreal, so I'm giving a talk in
Oh,
person.
okay,
So
okay.
so
Yeah, no, that's
that's why I
something
did not.
that's
Yeah.
good to look into though.
Yeah.
Yeah, thank you.
Because they are amplified with average like any PCR, any form of PCR, like the regular PCRs. I'm assume that must be the same case with the RD library prep because it is a kind of a PCR, and just because of the sequence similarity, they get amplified along with the mitochondrial counterpart.
Yeah, no, that absolutely makes
Yeah.
sense. Yeah,
Okay.
I'm gonna look into that. Thank you for that insight.
Okay. All right.
All right, anyone else? So if we have no more questions, you have an hour and a half to grab a meal at whatever meal is appropriate for your time zone. And we will see you back here about an hour and a half from now.